Hi there, science fans. Welcome to another Science and Other Stuff for Senior Citizens video. This video will introduce you to the concept of a recent therapy developed to treat a variety of cancers. This concept will be presented in two sequential videos. This first will be of a more general introduction to the science of immunotherapy, and the second video will be more in-depth presentation of the biology and the mechanisms associated with the utility of this therapy. I was stimulated to develop this series because of the vaccines developed and produced to prevent infection by the novel COVID-19 virus. The mainstream media and government institutions like the CDC and FDA have bombarded us with terms such as messenger RNA, innate and adaptive immune system, cytokine storms, etc. Their presentations have been informative but not very scientific. We are left with the complex terminology and wonderment of the so-called instant development of these magical medical solutions when in fact much of the development has been in research pipeline for at least 10 years and in some cases 50 years. So cancer immunotherapy solutions are dependent on these new technologies, and the case of messenger RNA vaccines have been at the forefront of utilizations of these powerful new technologies to solve previously unattainable goals in cancer therapies. This slide introduces the outline of today's presentation. Since each of these topics are very complex, I will present only an introduction for each of them. The subsequent videos will present the immune system and cancer immunotherapy in more detail. And if you have specific questions, or if you wish to have more detail on one or more of these topics, leave me a comment in the comment section. So what is cancer? Cancer is a disease in which some of the body's cells grow uncontrollably and may spread to other parts of the body. This latter event is termed metastases. In the cartoon, Panel A presents an illustration of a section of an organ, where the cells are contained and their morphology or individual shapes are defined and orderly. The different normal cells are also contained within a basement membrane, separating one tissue from another. In Panel B, the abnormal cells have very different shape and are increasing their number via uncontrollable cell division. Additionally, the basement membrane no longer acts as a barrier, and the abnormal cells are invading the surrounding tissue, beginning the process of metastases to other organs. This process is facilitated by passage into blood vessels. There are several types of cancer, including cancer of the blood and solid tumors. Blood cancers affect blood cells and the bone marrow. There are three major types of blood cancer: leukemia, lymphoma, and myeloma. Within each of these categories, there exist subtypes of blood cancer. Since identification of these blood cancers only require a sample of blood or bone marrow, these types of cancers are more readily detected than solid tumors, which require the use of more invasive procedures. Solid tumors include cancers of the lung, breast, prostate, colon and rectum, and bladder. Solid tumor cells are not present in large enough numbers in body fluids to be detected with a blood test. Although these two types of cancers have very different architecture, they display similar traits at the cellular and molecular levels. These traits include isolated abnormal cells, genetically unstable cells, and disordered differentiation. This slide graphically illustrates the normal cells shown in Panel A and the abnormal or cancer cells shown in Panels B and C. Panel A represents a normal cell, indicating the location of DNA in the cell nucleus. The structure of the DNA is highly coiled in a chromosome. The human has 23 pairs of chromosomes in each cell's nucleus. One pair is contributed by the father, while the other chromosome pair is contributed by the mother. During different phases of the cell cycle, the chromosomes unwind to replicate the DNA, doubling the DNA content for eventual distribution of half of it to each daughter cell. If you would like a video developed to describe this process, leave me a comment in the comment section. Also in Panel A gene is represented by a section of the DNA helix. Information for all proteins required for a healthy living cell is contained in the approximately 30,000 genes located in every human cell. These genes are expressed at different times during human development, that is, the embryo, fetus, after birth, early childhood, and adulthood. This normal gene expression results in what we call a normal human containing about 30 trillion cells. This normal human began as a single cell, the fertilized egg, and because of the faithful replication of DNA and the normal expression of that DNA during different parts of human development, a normal individual is produced. While each of the approximate 30 trillion cells have the same DNA in them, different cells such as brain, liver, heart, hair, blood, etc., express those genes at different parts of human development, resulting in the final developed organ. For example, brain cells and liver cells have similar DNA, but very different functions. The process of ordered differentiation results in a normal human. However, if a certain set of normal genes are mutated because of genetic inheritance or because of exposure to a chemical that causes mutations, disordered differentiation occurs and may result in a human predisposed to cancer. Panels B and C show cells that have mutated genes that predispose an individual to cancer. BRCA1 and BRCA2 are cancer genes that can predispose an individual to breast cancer. If you would like a video with a more in-depth discussion of oncogenes and suppressor genes, leave me a comment in the comment section. This slide and the next slide present some major differences between normal cells and cancer cells. You can read those differences. If you would like a video expanding on the differences between normal cells and cancer cells, leave me a comment in the comment section. Medical treatment of patients for their presented illness has been a one-size-fits-all approach to drug development and prescribing medications as depicted in the upper panel of this slide. Patients with headaches, fevers, cancer, or other ailments were generally treated in the same way. Patients suffering from mental illness were also treated with a one-size-fits-all approach. Blood and solid tumors were either treated by similar radiation, chemotherapy, surgery, or some combination of these treatments. However, with the advent of new molecular technologies developed during the past 20 years, medicine is evolving to a more personalized or precision approach as depicted in the lower panel in this diagram. Patients may present with similar symptoms but are treated in very different ways. Individuals with very different capacity to metabolize a specific drug or class of drugs would be prescribed different doses of that drug to maintain an effective level in the blood for a period of time. Some individuals may not be able to tolerate a drug based on their genotype or genetic makeup of that individual, so a different drug would need to be prescribed for that person. Personalized medicine can be achieved because of recent developments in genomic, proteomic, and metabolomic technologies. These tools will be discussed in the next slide. Research over the last 10 years has provided medicine with a clearer understanding of how the human immune system functions. This understanding allows physicians the ability to prescribe drugs that can boost the individual's immune system to fight and conquer foreign invaders such as viruses and bacteria, as well as their own cancer cells. Immunotherapy is a tool that will be described in a later slide, and a subsequent in-depth video will be dedicated to that amazing tool. This slide introduces us to the genomics, proteomic, and metabolomic technology that allows us to practice personalized medicine. We can readily isolate DNA from any individual. Sometimes white blood cells are all that's needed. Upon isolation of the DNA, its sequence can be determined, or sometimes a partial sequence is all that is needed. Sequencing determines the order of the letter A, T, G, and C, which dictate the kind and function of the specific protein produced. Sometimes identification of an individual's specific genetic mutations or polymorphisms are enough to understand how to treat an individual. Sometimes isolation and sequencing of the dictated protein is required. So specific tools have been developed to isolate, sequence, and determine the protein's function, allowing treatment at the personal level. Generally, the important protein is an enzyme which catalyzes or causes a change in a substance to another substance that can be measured in the body fluid using metabolomic technology. At present, all of these tools are available and are being evaluated as to their usefulness in prescribing medication for the individual and not the population. Many specific human pathological pathways are very well understood, and genomic approaches are being applied to treating patients presenting symptoms of those pathways. If you would like a more in-depth video discussion of these new tools, leave a comment in the comment section. The immune system protects our body from outside invaders, such as viruses, bacteria, fungi, and toxins that are produced by microbes. It is made up of different organs, cells, and proteins that work together. The panel on the left side shows the various location of organs that are part of the immune system. Some of these organs, like the bone marrow, are where immune cells are born. Other organs, like the thymus, act as schools, where an immune cell matures to be a more potent soldier to help fight the invaders. In panel B, we are introduced to the innate immune and adaptive immune systems. The innate immune system is made up of the rapid response system. This system patrols the body and is the first to respond when it finds an invader. When an invader is recognized, certain cells of the innate system surround and engulf the invader, and it is killed inside of the cell. The acquired immune system contains certain cell types that produce antibodies that can inactivate the invader, and other cells that memorize the quality of the invader, so upon further invasion by that pathogen, the adaptive system can more rapidly respond and inactivate the invading pathogen. If any of you would like a more in-depth presentation of the immune system, leave me a comment in the comment section. Immunotherapy is a type of therapy that uses substances to stimulate or suppress the immune system to help the body fight cancer, infection, and other diseases. This cartoon on the left describes the basics of immunotherapy. The cells of the immune system detects the cancer cell as non-self and proceeds to kill it. The detection relies on certain non-self or cancer proteins covering the surface of the cancer cell. Our immune system recognizes these cancer proteins or foreign antigens and initiates a cascade of events to destroy those cells. Sometimes the cancer cell tries to trick the immune system by hiding their foreign antigens and may not be readily recognized by the immune system. So researchers have developed at least 3 types of immunotherapy to fight cancer: nonspecific stimulation, T-cell transfer therapy, and immune checkpoint inhibitors. Use of the T-cell transfer therapy turns the therapy into a personalized medicine tool. This methodology uses genetic engineering of the individual's own immune cells to destroy the cancer from progressing to a point of no return. The next video in this series will present an in-depth presentation describing cancer immunotherapy, its importance in our cancer treatment toolbox, and how this therapy uses the patient's own cells to fight their cancer. Hope to see you then. Don't forget to subscribe and ring the bell to be apprised of Cancer Immunotherapy Part 2 and for other videos on science and other stuff for senior citizens.